we start with the laboratory diagnosis, the first thing that you have to know is sampling. Where will you take the sample from? Now, when I talk about sample, I've got a long list because I can never predict where the fungal infection has occurred. Now, when I talk about the usual sites, yes, hair, we all know hair, skin and nail. Dermatology is going to be very famous. So these are the samples that will come to you. Fungal infections can come to you with pus. So maybe they've given you a pus sample, a few ml of pus has come to you for examination. Lung is a very common site. So if there is a pulmonary involvement, the most common sample that I'm expecting is sputum. If sputum is not available, they might have given me bronchial washings or a bronchoalveolar lavage. And let me tell you, there is one infection in which sputum is not formed. That person is not going to have sputum. That person is going to have dry cough, and that infection is going to be pneumocystis carinii. Pneumocystis carinii, and pneumocystis carinii is definitely going to come. How will you make the diagnosis? If there's no sputum production, you're going to go ahead with a bronchial washing or a bronchoalveolar lavage. Okay, lastly, if nothing works, then you need to go in for a lung biopsy, which is a little rare thing to do, can be done in cases of certain fungal infections, which I'll talk about in a while. Other than that, there can be blood sample. If there's a meningitis case like cryptococcal meningitis, you'll get a CSF sample. If there's urine or any body fluid involvement, you'll get a sample accordingly. So all said and done, what we mean by this is that depending on the site of or the organ affected, you're going to get the sample. But yes, repeating, some of the most important samples will be hair, skin and nail. Moving forward, what are the different techniques by which we do the detection? So please remember, the first one is going to be the direct examination. Means you are going to just pick up the whatever hair, skin and nail is there, and you're going to look at it under the microscope. Do you know how do we do a direct examination? It is done by something known as a KOH mount. I'll first start with this. I'll first start with a KOH mount. This is how the picture of a KOH mount is going to look like. So first and foremost, what is KOH? KOH is not any stain. It's not any pink, blue, green color stain. KOH is potassium hydroxide. And how much percentage am I using? I'm using a 10 to 20% of potassium hydroxide. Reason, why am I using this? And I can see the picture also. I'm not getting any color. It's a totally transparent kind of a slide. Please remember, this is used because K for K, this is going to digest keratin for me. Potassium, KOH, is going to digest keratin. Why do I want to digest keratin? What kind of samples am I dealing with? I'm dealing with samples like hair, skin and nail, right? When I'm dealing with hair, skin and nail, these samples are the ones which are very, very rich in keratin. Now, if they're very rich in keratin, just imagine a nail. A nail is going to be so thick. How can you just put a nail under the microscope and expect that you'll see a fungus growing inside it? No, you'll have to remove all the keratin from the nail. You will have to digest the keratin. So see, you digested the keratin and then you started seeing beautiful hyphae. Beautiful for us, obviously not for the patient, but yeah, you can see these long beautiful hyphae that are there, but you could only see that once the keratin was removed, and that is exactly what is done over here. Well, having said that, this is about the KOH mount. That was direct examination without putting any colors. The second thing that you'll do is, first you'll start putting some blue, green, pink colors. So let's start with all the stains that we have. The first one, that is quite a famous one, you can suddenly see that the fungal hyphae have become all black, right? So this stain is known as the GMS. This is known as Gomorrhees or Grocott methenamine silver stain. This is known as Gomorrhea or Grocott methenamine silver stain. Now I'm going to tell you, and I have told you multiple other times also in your path and microbiology lectures, that whenever the word silver comes up in a particular stain, the color that you are going to get is always going to be a black color. So that's a trick which will help you solve so many other things. You must have heard in, do you remember, let me do a very quick recap. Do you remember in your pathology lectures in melanin, have you done a melanin stain, M for M, melanin, Mason, Fontana, silver stain? Again, Mason Fontana silver stain gave what color? It gave a black color. So whenever, and I ask you one more thing from bacteriology, what is the stain for Helicobacter pylori? For Helicobacter pylori, I hope you've been through the bacteriology lectures. Warthin starry silver stain. Again, Warthin starry silver stain gives what color? It gets you a black color, because anything with the word silver is going to get you a black color. So here we have Gomorrhea or Grocott methenamine silver stain for fungus, that gets you a black color. So this was the first stain that we had. The second one that we have is a very usual one. Suddenly they've become all pink. The hyphae have become all pink. So what do I need to know? Please remember P for pass and P for pink. And what is pass? If you want to know the full form, pass stands for periodic acid Schiff. If you're not learning the full form, you're not missing out on much. So let me tell you, periodic acid Schiff is P for P. Pass is going to give a pink color. Coming to the third and the fourth, which I want you to first read and then I want you to ignore. The next two that we have are going to be, there's something known as the gram stain. Gram stain is something I usually do for bacteria, right? Gram positive, gram negative. But please remember, for fungus also, and what is the color? With the gram stain, I'm getting what color? I'm getting a purple color. I'm getting a purple color. This means fungal organisms are gram positive. Fungal organisms are gram positive. Coming to the fourth one, and that is what we have, something known as the very famous. Can you finish micro and path without discussing the famous pink and blue? That is the HNE stain, right? So please remember HNE, you can see all the fungus has become pink and blue. So that is the HNE stain. Well, having discussed it, so all in all, when I go back, there is a black color stain called the Gomori methenamine silver. Then the next one is going to be the pass or the pink color. Then we have the third and the fourth one, which I told you, read and forget. So what is gram stain? Gram stain is going to give you a purple color, gram positive, and there's an HNE stain. Finally, coming to the fifth one, and the fifth one is known as CFW. What is CFW? It is calco flower white. Now, there is a very interesting mnemonic behind it. Please remember, when I talk about calco flower, the flower word will remind you that this is a fluorescent stain. See, the flower word is reminding you that this is not a regular stain, this is a fluorescent stain. So, can you now see the background is all black? I've got you a black background because it's all dark, and what is going to come up in front of it is something shining. Something is shining. So why is something shining? Because you've used a fluorescent stain, right? But having said that, did you also notice something, that the entire fungus has not become shiny? Only the outline over here and the budding, only the outline part of it over here and over here is becoming shiny. Why so? Outline means what? Outline means cell wall. And have I already told you that what is the cell wall made up of? Go back to the first sentence of this chapter. The cell wall is made up of chitin. So remember, calco flower white binds to chitin. So now the mnemonic is complete, which means the C of calco will remind you of C of chitin. It binds to chitin, that is only the cell wall part. And the floor word will remind you of fluorescent stain. So calco flower white is a fluorescent stain. Coming to the sixth stain that we have, and that is now sixth and seventh. Now let me tell you, there's a very famous organism which you will definitely get a question on, and that is Cryptococcus. And the question will always be related to cryptococcal meningitis because that's the most common organ that it affects. Now, having said that, please remember, cryptococcal meningitis, a meningitis patient coming to you, what sample will you be taking from the person? We'll be taking a CSF sample. Now, when I talk about a CSF sample, the stain that I'm going to put is a negative stain. First, let me give you examples of it. Negative stain is going to be what? It's India ink and nigrosin. There's a stain called India ink and there's a stain called nigrosin. What do you need to know about these? Why did I call them negative? And the same concept I have taught you in your general microbiology also. Why do we call it negative? Because look at the background. It's actually the background that you are going to stain. The background is stained, and what is not stained? Look at this. What is this white thing coming? Something has not picked up the stain. That is the capsule of Cryptococcus. That is the capsule of Cryptococcus. That is what I wanted to tell you. Cryptococcus is a capsulated organism, and that capsule is very, very weird. You know, you try to give it any pink, blue, green color, capsule doesn't take any stain. It doesn't take any color. As a result of which, what happens is you will have to do the opposite approach. So I don't know whether you guys are pretty young and don't belong to our generation, but long back when you didn't have too many phones to click photographs, we used to take photographs with a camera and a Polaroid, and we used to have a reel, and the reel used to then print print out a lot of negatives. Maybe you've seen that with your parents. Makes me feel a little old while I say that to you. But yeah, negatives, the concept, I'm sure everyone's seen it. The concept of negatives was that the background was dark and the person's photo was coming highlighted out, right? The same thing is happening in a negative stain. That is, the background is coming out to be dark, and what is highlighted is the cryptococcal capsule. That is, if you received a CSF sample, that's the trick over here. What if the person had a biopsy done? Not a CSF sample, but what if the person had a biopsy done? So please remember, if you will receive a biopsy like cryptococcal meningitis or brain biopsy, or maybe cryptococcus has affected the lungs, so lung biopsy. If you ever get a biopsy, then what stain will you use for cryptococcus? Then for the same cryptococcus, I won't be using India ink. I will be using this nice rose, reddish, pinkish color stain, and that is known as mucicarmin. This stain is referred to as mucicarmin. So that is the 7th stain that you have done. So remember, for cryptococcus, reading what sample has been given to me is very important. If they have given you an India ink or a negative, or they've given you a CSF sample, then you go in for India ink or nigrosin. But if they've given you a biopsy sample, then you go ahead with mucicarmin. Having said that, these were all the different stains that you had. So coming back, first you did direct examination with KOH, then you went ahead with stains. Let's do a quick recap. The black color stain was Gomori methenamine silver. The pink color stain was P for PAS. So PAS was positive. For cryptococcus, we had the negative stains if you were talking about the CSF sample. For cryptococcus, you had mucicarmin if you were talking about the biopsy sample. And 2 miscellaneous ones were gram positivity and HNE stain. So that wraps up all the stains for us. Now coming to the third one, and that is a culture, and this is a previous year question, very, very important for all of you. So what is the culture that we use? The very famous Sabauroud's dextrose agar, that is SDA. Sabauroud's dextrose agar, or SDA, let's dissect it, is obviously going to contain dextrose, there's no doubt about it. The way you write SDA, the question that has come in your previous years is that what is the pH at which you are going to deal with it? So remember, the way you write S, it either looks like a 5 or a 6. So I've clubbed it. It is a pH of 5.6. The basic concept is that they ask you that this is an acidic pH. Okay, we require an acidic pH. Repeating once again, the way you write SDA is going to be a 5 or 6. 5.6 is the pH that is there. Apart from that, I have noticed that SDA has antibiotics also. What antibiotics? Gentamicin, chloramphenicol have been added. Why? So that it can inhibit or kill the surrounding bacteria. For the killing or the inhibition of the bacteria, I have added antibiotics. Apart from that, we can also have chlorheximide, cycloheximide added. Why do we have cycloheximide added? So please remember, you also want to finish off other saprophytic fungi. You don't want any saprophytic fungi to grow. You only want your pathogenic fungi to grow. So you basically want to get rid of 2 things. Number 1, you want to get rid of the bacteria. So for bacteria killing, you are going to give antibiotics. Second, you want to get rid of the saprophytic fungi. So for that, you're going to have cycloheximide. So these are questions that you get. Which antibiotics are you using? Gentamicin and chloramphenicol. Which, agent for killing the saprophytic fungi are you using? Cycloheximide. What is the pH? Something acidic, 5.6. And obviously the name has dextrose in it. So Sabauroud's dextrose will obviously have dextrose in it. Okay, having said that, now the next technique that I'm going to do is I'm going to take a little bit of this culture with a loop, and I say I want to look at it under the microscope. That is the next thing that I desire to do. So what am I going to do? Repeating once again, I am going to take a little bit of that sample from here and I'm going to look at it under the microscope. Whenever I look at anything under the microscope, I need a stain, and that is a blue color stain called lactophenol cotton blue. What do we need to know about it? So there are, as the name suggests, lactophenol cotton blue. So when I say lacto, means lactic acid has been used. Why has lactic acid been used? Lactic acid is going to help in preventing or preserving the morphology of the fungus. Lactic acid is going to help in preserving the morphology of the fungus. Okay, when I say lacto, then next is phenol. So why have we added phenol? I think everyone knows phenol is a disinfectant. Yes, so you want all the other miscellaneous things to be killed. So yes, you're adding a disinfectant along with phenol. We also have glycerol. Glycerol is something, the name doesn't have it, but please remember, glycerol is something, glycerol adds a little bit of water. When I say that it adds a little bit of water, it is something which is going to be hygroscopic. That is the description given to it. When I say hygroscopic, means anything that is going to prevent drying. Anything that is going to prevent drying, because it's providing that hygroscopic, that water ability, that is glycerol. So remember that, because it's not in the name. We only call it lactophenol cotton blue, but it also has glycerol. And finally, you have cotton blue. What is cotton blue? Cotton blue is the dye. Cotton blue is the one that is actually going to add the color to it, the blue color, and you can see that under the microscope. Finally, the color that you've got is a blue color. So let's repeat. When I say lactophenol cotton blue, lacto is for preserving morphology, phenol is a disinfectant, we studied it. Glycerol is another thing that we have, which is going to give all the water and prevent the drying, and cotton blue will give the color. So tell me, are you putting lactophenol? Now comes the real question, so make a table around it. Are you putting lactophenol cotton blue directly onto the sample? No. You are first growing the sample on a culture plate. From the culture plate, you are taking the sample and putting lactophenol cotton blue. So it is used for studying culture morphology, and that has been one of the most famous questions of the previous years. What is the stain used for studying culture morphology? It is lactophenol cotton blue. Well, having said that, almost coming towards the end of the culture media. So the main culture media was obviously SDA. But if I do a little bit of twists and turns, there is another culture media called the corn meal agar. And now I'm going to tell you the story of the 3 C's. So when we talk about corn meal agar, you know in which situation will you use it? When I want to grow chlamydospores. When I want to grow chlamydospores, that is exactly when I use corn meal agar. And I've already told you chlamydospores are grown or are grown by which particular organism? Candida. So repeating once again, when I'm talking about corn meal agar, I grow chlamydospores in it, and which organism shows you that? Candida shows you that. So that's another agar that you have to know. Then, because we are talking about Candida, I'll add another C onto that Candida, because Candida is also grown on another agar known as the chrome agar. Candida is also grown on chrome agar. So that will tell you chrome. Chrome means what? Multicolored. You're going to get an agar with a lot of colors. So again, C for C. Chrome agar is something that you grow. Candida, and you can see Candida tropicalis, albicans, cruzi, so many other things written. Do you have to learn these colors? No, you don't. You just have to know that yes, there is a Candida, and that Candida is basically grown on this colorful media. Okay, coming to the last one, suddenly everything's going to become black. So you either call it a birdseed agar or you call it a birdseed Niger agar. And why so? Because Niger word is going to tell you about that brownish blackish color. And this is very famous. I'm not telling you the pathophysiology behind it right now, but this is very famous for Cryptococcus. How to learn this? What is going to be the mnemonic? What is the story of Cryptococcus? We'll come to that. It has something to do with birds. So yes, you will be able to learn it very, very easily. But for the time being, remember, if I see a brownish color Niger, brownish color agar, that's a birdseed Niger agar seen with Cryptococcus. Well, having said that, if we want to do a very, very quick recap of whatever we have studied up till now, and that is something that I've introduced in these lectures, because I feel that there are some topics which are very image heavy. So while you study, you feel you've analyzed it well, but towards the end, you obviously feel that you need a quick summary of a minute or two so that everything that you studied in the past 15-20 minutes can be summarized quickly. This is what is going to help me. So if I show you all the pictures of what we have studied up till now, what is that modality which is going to give you that black color? I think everyone knows black color is going to be with gomori, methenamine, silver stain. So like I said, the black color is always going to be something with silver. So gomori, methenamine, silver stain. Okay, coming to the next, suddenly it becomes everything pink. So P for pink, and you're going to get what? P for pass. So we are going to now label it as pass. P for pink, P for pass. Coming to next, something is becoming fluorescent. As soon as we say fluorescent, we are going to use calco flower white. Remember, flower had the word fluorescent, calco flower white. Look at the next three. The next three are for the same organism, and that is Cryptococcus. What do we need to know about Cryptococcus and this? The first one must have been the CSF sample, because I'm using negative stain. I'm using India ink negative stain. The second one must have been a biopsy sample, because I have something red in color, and that is going to be mucicarmine. And like I told you, what is the agar? Something to do with birds and black, right? So there is a bird seed Niger agar. There is a bird seed Niger agar that we have, that sums up the entire story of Cryptococcus. Okay, what is the usual agar that we use? So yes, the first picture out here, the first picture is going to be the SDA, right? The Sabauroud's dextrose agar. Remember what was the pH? The pH was 5.6. And from that SDA, if I want to grow something as a culture morphology, so for culture morphology, the blue color stain that we use is lactophenol cotton blue. Finally, coming to the colorful media, which is the one that requires a colorful media. So this is known as the chrome agar. Chrome because it has so many colors, and C for C, chrome agar is very, very important when it comes to Candida diagnosis. So yes, this is one slide which wraps up everything that you have studied up till now in this chapter in the most concise manner. And before I make you practice a few questions, please remember, coming all the way back, you have done the culture, but there are some other things also that you can do. You can do antibody testing. That is one thing. You can do skin tests, which are not very commonly done, but like you had skin test for TB, skin test for leprosy and others, similarly for fungal organisms also you have skin tests. And lastly, something that you can do possibly anywhere starting from bacteria, virus and then fungus also is a polymerase chain reaction. So nothing very fancy about these three things that I had to tell you, but yeah, the direct examination with KOH, the stains and the cultures, those are the most important. Let's do a quick recap, and also now let's practice one or two questions which might help you. So that is a question that is going to help you now. There's a patient who's presented with scaly skin, redness, irritation and small blisters all over his skin. The doctor suspected fungal infection and sent his skin scrapings to the laboratory. Which culture media is useful for culturing the fungus? So firstly, even if you would have read the last sentence, which culture media is used for culturing the fungus, you know for a fact that for fungal organisms, the culture media is SDA. But obviously, the purpose of this question is not this. You have to tell me the others also, because by now I'm assuming you've heard throughout the bacteriology section also. So when I talk about potassium tellurite agar, do you all remember this was a selective media? This was a selective media that you had for what? This was a selective media for Corynebacterium diphtheria, right? Yaad aa gaya? Okay. When we say triple N, for triple N I had told you NNN media is used in two things. Number one, it is Leishmania, that is Kala Azar, and the other one is going to be Trypanosoma cruzi. So I hope you remember Leishmania was the organism causing Kala Azar and Trypanosoma cruzi was the organism that was causing Chagas disease, and for both of these we had NNN medium. Lastly, coming to chocolate agar, can be used in so many things, but one place where it was important was Haemophilus influenza. Do you remember that concept of satellitism? So yes, Haemophilus influenza can definitely be grown on a chocolate agar. So over here, because fungus was asked, your answer is simple. It's going to be SDA. Coming on to another question that we have, which of the following is not dimorphic? First, you will have to learn and recall which were the dimorphic fungi, and now you will be learning which of them is not dimorphic. So please remember, we had a mnemonic, right? Body, heat, probably changes shape. Can you do a quick recap with me? B was for blastomyces. H was for histoplasmosis. P is for Penicillium marnefi. So something to do with marnefi, Penicillium marnefi was mentioned. Next we have paracoccidioidomycosis. Then we have coccidioidomycosis, and then we have Sporothrix schenki or sporotrichosis. So what do we have to know? Now once again, Talaromyces marnefi. See, whether you call it Talaromyces marnefi or Penicillium marnefi, it's the same. So is this a dimorphic fungus? Yes. Coming to Sporothrix schenki, sporotrichosis, is that a dimorphic fungus? Yes. Histoplasmosis or histoplasma capsulatum, yes. So which of them is not dimorphic was the question, and not dimorphic is Cryptococcus neoformans, because I think everyone remembers Cryptococcus was the yeast form. When I say yeast form, means Cryptococcus was the one that was showing me budding. So the answer over here is option B. Yes, with that we wrap up our discussion of the introductory part of mycology. Now we are going to start off with the next